So today I'm going to present this paper, Enhanced Tonic GABAA Inhibition in Typical Absence Epilepsy. It's out of a lab from Vincenzo Crunelli, who, for the sake of full disclosure, I worked with Vincenzo for 5 years. I lived in David Cope's house, and Adam Errington's a personal friend of mine. So, I'm a little bit biased, but on the other hand, that means I do know really what's going on here. Um, So we've already learned about absence epilepsy, right? So it is um, this epilepsy disorder that involves brief loss of consciousness, and it's sort of its hallmark are these spike and wave seizures, or these spike and wave EEG discharges. Um, it's linked to something going wrong in the thalamus, but we know that the fundamental problem is something in the cortex, and something that I may not have mentioned earlier is that it's one of the most common pediatric epilepsies. While we know maybe more about it than some other epilepsies, we still don't really know what causes it. It's associated with mutations in T-type calcium channels, but also GABAA receptors. And we also know that some drugs that enhance GABA function seem to make absence epilepsy worse. And we also know that spike and wave discharges is something to do with the thalamic reticular nuclei, which is this inhibitory shell of the thalamus. So, people have wondered that, or hypothesized that all epilepsies are to do with inhibition being ineffectual, right? Because if you block inhibition, seizures happen. If you have somebody with seizures and you give them something that enhances inhibition, the seizures stop. It just kind of makes sense. But no one has found good evidence for it outside of a few specific genetic epilepsies. Now, we typically think of inhibition as, we have inhibitory neurons that release GABA. They have vesicles containing GABA. They release those vesicles. The postsynaptic cell is inhibited. And this is what is called synaptic or phasic inhibition. Phasic in that it is on for a little phase and then it disappears. But in the mid-90s, it became clear that there is another way that GABA acts, that all of this GABA gets released, and some of it ends up being sensed by high affinity GABA receptors that exist outside the synapse, and that they just are constantly activated a little bit by the GABA that's floating around. So these are extrasynaptic receptors that generate something called tonic inhibition. It's tonic, it's constant, right? So they're always providing a little inhibitory signal. Now, much like in my office, there is a fan in the roof. I don't notice it all day long, but at 5 o'clock it turns off, and when it turns off, the silence is deafeningly pleasant. And tonic inhibition is much the same. It's always there, right? So you can't really tell anything about it. But if you block GABAA receptors, as you see here, then you see the current disappear. So the current was, in this case, was downwards, and when you block it, the current goes up the page. Sorry for the yawning. Um, so, although I should add, in some figures I'm going to show the tonic current's going down the page. It depends on whether there's lots of chloride inside the cell or not lots of chloride inside the cell. But the point is, the way that you reveal the magnitude of the tonic current is to block it, and then you see the change in current induced when you block the GABAA receptor, and then you know the size of the tonic current. Obviously, this is what phasic inhibition looks like. This is synaptic inhibition. This is what you see here. GABA gets released, binds to a synaptic receptor. You get the current starts to flow, and then the GABA is released from the receptor. It's taken up by things like this GAT1, the GABA transporter 1, taken back into the presynaptic terminal. Also, astrocytes also express GABA transporters, primarily GAT3. Apologies again for yawning. It's Friday night where I am. Now, just to explain some drugs that are going to be used. So you may see SR95531, also called gabazine, used. There's a drug called THDOC and another one called THIP. And these are both drugs that enhance extrasynaptic GABAA receptors especially. So here you see the normal current, they apply SR95531, the current goes up, so the difference between there, here and the dashed line is how much tonic inhibition there was. And as they apply this THDOC, then that tonic inhibition is larger because there's a bigger difference when they block it. Now, here is sort of an important one. What they say is the delta subunit is critical to the tonic current. So it's not super clear in this figure. This is just an important initial paper. I used to carry this paper around with me for a long period of time. So they've got a mouse that's lacking the delta subunit, and when they apply gabazine, you still see that there is a small change in the tonic current. But if we look at the scale, it's a couple of picoamps. While if we go back here, it used to be about 20 picoamps. So we've massively reduced it, and we've reduced the effect of THDOC is now nothing, right? So before, when we used 100 nanomole THDOC, we had 40, 50 picoamps. So at least in the cerebellum, the dentate gyrus, it is this delta containing GABAA receptors that mediate the tonic inhibition, or the vast majority of it. So anatomists already knew actually that the delta subunit was found outside the synapse. They just hadn't quite put two and two together. So here we see an electron micrograph. Here's the presynaptic terminal. You can sort of make out the vesicles that are in it. Here's the presynaptic density, or the postsynaptic density, sorry. And here you see a electron dense gold dot caused by a delta receptor, and you can see it's right at the edge, not massively outside the synapse, is worth pointing out, right, but at the edge of the synapse. So here we see the tonic current going in the other direction, and so we already knew it was mediated by delta containing GABAA receptors. Now this paper was one that showed that it was alpha 4 containing receptors, telling us it's the alpha 4 delta containing receptors, and we already knew that basically alpha 4 must have a beta subunit as well. So this receptor is alpha 4 beta something delta. And you can see there's a couple of important points here. One, here is just in a normal mouse, they add NO711, which blocks GABA transporter. So there's more GABA floating around, and you can see the current goes up. They put in a higher concentration, the current goes up, and then they apply picrotoxin, which is a classical GABAA antagonist, and they get the reduction. So what they're saying is there was 16 picoamps of tonic current in this one, but you can see it was the cell, but it was increased by blocking the GABA transporter. While in the alpha 4 knockout mouse, NO711 had very little effect, maybe a slight effect by this higher concentration, and then when they put on picrotoxin to block all GABAA receptors, there was only sort of 2 picoamps of tonic current. Turns out in the cerebellum, in the cerebellar granule cells, it's the alpha 6 beta X delta receptor, but still the delta subunit is just the alpha 4 versus the alpha 6. And just to sort of wrap the story up, in the cortex, there is also tonic inhibition. It's much smaller, and it is absolutely dependent on this alpha 5 subunit. So again, here's the wild type. They apply bicuculin, another GABAA receptor antagonist, and you can see the current goes up. Then when they wash the bicuculin away, the current returns. But in the alpha 5 knockout, nothing. And it's not just the alpha 5 delta, there's also the alpha 5 gamma that appears to play a role, though that's not demonstrated in this figure. So, first question. Is there tonic inhibition in the thalamus, right? So we're trying to ask this question: is tonic inhibition related to absence epilepsy? We know the absence epilepsy has something to do with the thalamus. Exactly what, we're not sure. So does the thalamus have tonic inhibition at all, right? Because if it doesn't, then there's no point in asking, does tonic inhibition in the thalamus have anything to do with absence epilepsy? And the answer is yes. So all of the primary sensory nuclei, here we have the LGN and the ventrobasal, so visual and somatosensory, and we see that there's a tonic current in both, but in the NRT, the inhibitory shell of the thalamus, there is nothing. And THIP, which is this delta subunit selective positive modulator, positively modulates this current, so we know it's mediated by delta containing GABAA receptors. So that's the backstory. Here we are now. So we know tonic inhibition is a thing. We know it's important. We know the thalamus has tonic inhibition. We know that inhibition can go wrong and cause absence epilepsy because there's a mutation in the gamma subunit of the GABAA receptor that causes absence epilepsy. And we know that the thalamus might go wrong in absence epilepsy. So does tonic inhibition go wrong in absence epilepsy? And this is the first figure, and in a sort of a common approach in high impact papers, this is really the most important figure in the whole paper, right? And what it shows here, right, is this is the magnitude of the tonic current by age of the animal. So P14, the animal's 14 days old, and you can see that the NEC is non-epileptic control, so it's the appropriate control for the gears rat. And you see that it's the same at P14, it's the same at P15, the tonic current's the same at P16, at P17, and then, and then at P17, there's a difference, and it keeps going at P18, P19, P20, P30, right? Now, what is absolutely critical about this is that seizures begin in gears rats on the same day in every rat, which is P16. So, on the day after the seizures begin, the tonic current is much bigger. Now, let's have a second, right? The tonic current is bigger in the animals with absence epilepsy. They are receiving more inhibition, which is sort of pretty unusual for how people think about epilepsy. It's worth repeating that this effect is seen in stargazer mice, lethargic mice, but not, despite me saying here, not in tottering mice, right? So what this is saying is that there's this huge correlation between, and I should be clear, stargazer and lethargic mice and tottering mice all have spike and wave discharges, though it's curious that in the tottering mouse, they all, the tonic current amplitude is high in both the tottering and the wild type. So maybe there's actually some mutations in the wild type control that we're aware of. Anyway, so we have this increase in tonic current when animals have seizures. It's even temporarily correlated, so it happens at the same time, but this is still correlative, right? So we do not know if this is causal at this point. Even more sort of curiously, we see that even pharmacological models of absence epilepsy induce an increase in the tonic current. So GHB is a recreational drug and a model of absence epilepsy. You give it to a mice, a rat, a cat, and they start having spike and wave discharges and behavioral arrest. And the tonic current amplitude is increased when you give this drug. Now that's very strange because GHB actually acts on GABAB receptors, but as I say here, I go on to show what causes this. Now in the paper, they claim that this result shows that the relationship between the tonic current and the absence of epilepsy is causative. Again, not again, this is not true, right? This does not show causation because they don't show that if you block the increase in the GABA-A tonic current, that you block the absence epilepsy. Okay, so what could be going on? What could be going on, right? So there are three sort of core explanations for what is happening here. That when animals have absence epilepsy, they release more GABA, so that makes more tonic current. They have more GABA receptors, that could make more tonic current, or less GABA is removed, and hence there'd be more GABA floating around, and hence more tonic current. So, could it be more GABA release? So what they do is they look at the rate of spontaneous inhibitory postsynaptic currents, and they say, look, they're basically the same. At P14, they're reduced in the gears, and at P18, they say that it's reduced in the gears. But other than that, across all other days, they're exactly the same. And this probably just reflects bad luck, statistical bad luck, as it were. This is not an absolute hard and fast proof, right? The problem is it's essentially impossible to measure the concentration of GABA that's floating around outside the synapse. So you always have to look at some kind of correlative measure. But the idea here is that if there's less synaptic events, then there's less GABA being released. And in this case, there's the same frequency of synaptic events, and hence the assumption is that there's the same amount of GABA released. More GABA receptors, they're going to skip over that one. But thankfully, because they do provide a mechanism, which is looking at GABA removal. So there are three GABA receptors. It's actually some really complicated nomenclature, but GAT1 is expressed on neurons, GAT3 is expressed on astrocytes, GAT2 may or may not even really be a thing, so GAT1 is blocked by NO711 and GAT3 is blocked by SNAP5114. And this is what they did. So they measured the tonic current size and again, this is in the ventrobasal thalamus. So this is in the thalamus, and we see that, as we knew before, here are the N numbers. So these are pretty impressive N numbers, 32. The tonic current is nearly doubled in the gears, right? But when they apply NO711 blocking GABA, well, you see the tonic current increases in the control, but there is no effect on the gear's epileptic rat, suggesting NO711 doesn't do anything. Now, when they apply SNAP5114, which blocks GAT3, they get an increase in the tonic current in both the control and epileptic rats, suggesting that GAT3 is just the same in both. And then when they apply NO711 on top of SNAP, again you see a massive increase in the tonic current in the wild type, because there's nothing to get rid of the GABA, but it has no effect on the epileptic rat. So even in this case, when there's tons and tons of GABA floating around, blocking the GAT1 has no effect, right? So it really seems like GAT1 has disappeared. Now, that was in the Gears rat, which is a sort of this gold standard model of absence epilepsy, but even in the Stargazer, that's not a great model of absence epilepsy, you get the exact same pattern. In the control, we see the increased tonic current. When you apply NO711, the tonic current goes up in the control, but not in the epileptic mice. When you put on SNAP, the tonic current goes up in both groups, and even in the presence of SNAP, NO711 has no effect. So the GAT1 transporter appears to be missing. Again, lethargic mice provide weird results. Now here, the lethargic mice do have an increase in the tonic current, and when they apply NO711, the tonic current actually got smaller in the epileptic animals. They don't talk about this result, unsurprisingly, because it doesn't even agree with some of their earlier ones, and it doesn't really make a lot of sense. But it's impressive that they would report a result that inherently is kind of nonsensical. You may think, well, hang on, shouldn't they have done it again or figured out what's going on? To me, it's more important that they were being honest than that they were working to make a perfectly airtight story. That's my take on that, right? Science sometimes doesn't work out like how you want it to, and reporting it as the truth is important. All right, so All of the results so far have been purely kind of correlative, and this is not really going to get beyond that, but it's at least going to take a bit of a different tack, right? So if we're saying that Gears rats and stargazer mice have absence epilepsy because their GAT1 receptor is missing, then that should mean that if we knock the GAT1 transporter out, we should get absence epilepsy. And indeed, you do. Now, the curious thing is the GAT1 knockout mice had been around for years, but nobody had ever looked or noticed or thought to check. And so when they put an EEG electrode in the GAT1 mice, they saw the spike and wave discharge. And more importantly, and this is, I think, really crucial, that the classical anti-absence drug ethosuximide, which doesn't stop tonic-clonic seizures, medial temporal lobe seizures, it only stops absence epilepsy, it basically completely cures the GAT1 knockout mice of their seizures. Now, what we show here is that, what they showed here is that they sort of did this same experiment as we just saw, but now pharmacologically. So they infused NO711 into genetically normal rats, and as soon as they did that, into the thalamus, I should add, as soon as they did that, the animal started having seizures, and then if they gave the animal ethosuximide, it stopped having seizures. So it really does seem like there is causation here, right? That if you block the if you get an animal with absence epilepsy, it has no GAT1 receptor and too much tonic, no GAT1 transporter and too much tonic current. And if you take a normal animal and block its GAT1, it develops absence epilepsy. They sort of really started hitting the sort of beating a dead horse here. So now they use THIP. We already saw how THIP enhanced the tonic current, right? And this is starting to be really causal here because previously all of these experiments sort of were showing that yes, we block the GAP1 receptor and there's lots of GABA floating around, but they weren't proving where the GABA was acting. But here they use THIP, which is selective for enhancing the tonic current, and when you infuse THIP into the thalamus, then the rat started having absence epilepsy, and again, you could treat it with ethosuximide. So you could stop the spike and wave discharges in these rats that were treated with THIP by giving them this classic anti-epilepsy drug, anti-absence drug. They were able to get even more sort of specific, or no, it's probably the same amount of specific, but they tell the story again, make this point again by showing that when you treated a mouse with GBL, which is basically GHB in a different form, you induced, you see here we have time spent in seizures per 15 minutes, and you can see that after the GBL injection in the wild type mice, they started spending a large portion of the time having spike and wave discharges, but in the delta knockout mice that had no delta receptor, GHB had very limited effect. Now they're really driving the causation home. So what they are doing is injecting antisense mRNA to the delta subunit. So this will essentially bind to the mRNA for the delta subunit and stop the delta subunit from being expressed. And what they showed in I was just trying to remember whether it's gears or stargazers, but I'm 90% sure it's gears, that when they infused this antisense mRNA, but not the missense mRNA, the time spent in seizures decreased, right? So they're able to essentially cure epileptic rats by injecting, by blocking the delta receptor in the thalamus. So what did they show? They showed that all models of absence, both genetic and chemical, have increased tonic GABA. they showed that novel ways to induce the tonic GABA current induced absence seizures, and blocking the tonic GABA current with antisense mRNA or genetically prevents absence seizures. So what this shows is that tonic current is sufficient and necessary for absence seizures. Or another way of putting that is that absence seizures occur if and only if the tonic current is enhanced in the thalamus, which is a real sort of landmark conclusion. What they didn't show is how, right? Um, how does the change in the tonic current come about? Like why um, does GAT1, I say GAT3 there, but why does GAT1 go away? And they don't really know, right? So this is basically them showing that in now this is in astrocytes. They apply GABA, and this current is the current caused by the uptake of GABA. And when they block all of the ion channels, um, they block all the transporters. They see no current. Um, but my hypothesis, and I think they still think this, not that they've been able to show it, is that it's some kind of activity-dependent regulation, right? Now, we don't know for certain that in mammals there's activity-dependent regulation of the GABA transporter, but in Drosophila we do, and in the fruit fly we do. So basically, if you activate GABAergic interneurons, then the GAT goes the GABA transporter goes up, and if you inhibit the GABAergic interneurons, the GABA transporter goes down, and that just kind of makes sort of homeostatic sense. You increase the amount of GABA, and the transporter is going to be over expressed. And it seems like this is caused by astrocytes sensing GABA via the GABA B receptor. And of course, that's of relevance because we know that GABA B agonists like GHB cause absence epilepsy. So we also, so this is basically showing that there, that activating the GABA B receptor enhances tonic current in mammals. So here we are using baclofen, or I'm using baclofen. This is a paper of mine, and when you apply baclofen, the tonic current increases, both in the thalamus and the dentate gyrus and the cerebellum. But one way or another, because we know that there's a cortical focus in absence epilepsy, it's probably that cortical activity does something to the thalamus, causing the tonic current to get bigger by decreasing the GAT1 receptor, GAT1 transporter expression. And then for some reason, this allows the cortex to entrain the thalamus and cause really detrimental absence seizures. So, it's always worth critiquing a paper, so they really didn't discuss any possible mechanisms in this paper. Now, I say it's not super their fault because they really had no idea, right? There was nothing to go on, right? There was no clues at this point in time to try and explain how this could be happening. I will say that there is some super weak and weird and undiscussed stuff in the supplemental materials. So this is them looking at the expression of GAT1 in the thalamus, and they basically say the GAT1 expression is the same. But yeah, they never mentioned this, right? So how is this happening? So we would assume that maybe there's some phosphorylation of GAT1 or some other modification that modulates how it behaves. But it would have been nice to for for them to maybe mention the fact that they couldn't actually detect a change in the expression of GAT1. And they really should have shown no change in GABA receptors, right? Despite them sort of providing an alternative mechanism that it's something to do with the GABA transporter, it would have been great for them to show that there was no change in GABA receptors. Sort of follow up, they've also been able to show, have also been able to show that you can essentially rescue, you can treat any of these genetic models by knocking out the delta receptor permanently, right? So in the paper they used antisense mRNA to knock down transiently the expression of the delta subunit. But what you can do is cross some of these genetic lines with a delta knockout such that the mouse has all of the genetic components that cause the absence epilepsy, but no delta receptor. And when you do that, these mice do not have absence epilepsy, really proving that you need that delta subunit to cause absence epilepsy.